Hi, welcome back to Discovering Patterns in the Microbiome. Today, in this video, we will be discussing clustering. Now, we saw this example a few videos back where there was a study that had discovered these three discrete types in the gut. And here's figure one, had these very nice looking separate clusters. There were 33 subjects in the data set, and the claim was that there were these three clusters. It turns out this visualization is actually a supervised visualization where the clusters were determined separately using a different algorithm, and then the visualization tries to make them look as separate as possible using the species information. So the more species you have, the more likely it is that random combinations of species will make the classes look separate. And so whether or not your clusters are valid, the visualization will look better and better. In other words, the clusters will get tighter and tighter the more dimensions you have in your data set relative to the number of samples. And so I have urged caution about using these types of supervised visualizations because they can lead you to believe that the clustering is actually stronger than it is. Now, when you're going to test for clustering structure, the one other pitfall is to use a relative measure of cluster strength only. So a common technique is to use the Kolinsky-Harabasz metric, which basically tells you which is more likely, 1 cluster, 2 cluster, 3 cluster, 4 clusters, and so on. But it doesn't demonstrate the existence. So sorry, it doesn't give you a score for 1 cluster. It just tells you 2 versus 3, 4, 5, and so on. So in general, it's better to have an absolute measure of clustering quality. For example, in this data set, we see that 2 clusters gets a low Kolinsky-Harabasz index score, 3 clusters gets a really high score, and then they start going back down. So clearly, if you're going to impose clusters on the data, 3 is better than 2 or any other value. However, we still don't know if there are clusters. And so another approach to doing this is the silhouette index. And silhouette index was devised as an absolute measure of cluster quality. So it gives a score between -1 and 1. So the silhouette index is basically you take for one cluster the average distance of its points to the nearest other cluster. Then you subtract from it the average distance to the same cluster, from one point to its assigned cluster. And so it's a measure of how far away something is, how far away other clusters are relative to the diameter of the current cluster. And the values that you get back tell you if it's a strong or weak clustering. So the proposed approach for doing this is that below 0.25 as a result means that there's no substantial structure. In between 0.25 and 0.5, the structure is weak, could be artificial. Anything above 0.5, there's some reasonable or even strong clustering structure. The silhouette index is actually somewhat low powered, so we've seen cases where it gives a low score when there is known clustering structure in the data or when other methods can detect it. Another method is the Dirichlet mixture model, which does do a pretty good job of telling you whether there is clustering structure at all. And then once there is clustering structure, it tells you a good job. It does a good job of telling you whether it's 2 or 3 or 4 and so on. However, there are cases where this also gives false positives. So you can have cases where if there are strong gradients of dominant species in your data, and you can have cases like, for example, in the human gut, where these gradients can cause the Dirichlet mixture model to produce a significant cluster structure when the data is actually just simulated. We know that there isn't a strong clustering structure. So there's a lot of controversy about how to use these. I'll give you my opinion at the end of the video on where I think it's appropriate. Another approach that makes a lot of sense to me that that I've used in a few data sets is called prediction strength. And prediction strength it's an alternative to the silhouette index. The way it works is you take whatever algorithm you're using for clustering, so it doesn't care what algorithm you use. You do clustering on half of the data. So you arbitrarily just cut the data in half, cluster one half of it, see what clusters you get. Then separately, you cluster the other half of the data, see what clusters you get. Then for each point in the second half of the data, B, you assign it to the nearest cluster in A. So all of this is premised on having a distance measure between all of the points or all of the samples. So again, in one half of the data, you assign each point to the closest cluster in the other half of the clustered data. And you compare the co-memberships of samples in the actual clustering in the B half of the data to the co-memberships that were obtained when you assigned points to their nearest clusters in A. So in other words, what you're saying is, if I take a random subsample of the data and generate clusters, how well do those clusters represent the clustering structure in the held out portion of the data? And the score that you report is the worst fraction for all of the clusters in B. So in other words, maybe in the first half of the data A, you were able to detect 2 clusters, but in the second half B, you detected 3 separate clusters. And so all of the points in, and let's say maybe some of the points in the new third cluster, when you assign them to their nearest cluster in A, some of them went to the first cluster, some of them went to the second cluster, so they got split up. And maybe in that case, only half of the co-memberships of that cluster were retained when you mapped them onto the clusters from the first half of the data. And so in that case, prediction strength would be 0.5. A couple of years ago, with some collaborators, we applied prediction strength to a number of different data sets, including simulated data, and found that it worked pretty well. So for example, here's a data set simulated to have 4 distinct clusters, and then I have prediction strength, I have silhouette width, and I have the Kolinsky-Herabasz metrics. All 3 of these metrics show a peak at 4 clusters. You see 2 clusters is worse, 3 clusters is a little bit better, 4 clusters is best, and then it starts going down again. But only prediction strength here gives a qualitative score, a quantitative score for the absolute quality of the clusters that tells you that there are indeed clusters. So, so prediction strength for this data set is almost 100%. The average silhouette width tops out at about 0.3, so it's actually underpowered for detecting these clusters. And then the Kolinsky-Herabasz metric also has a peak at 4, but it's not telling you the overall quality. It's not saying, yes, the clusters are real. We applied this to different body sites from the Human Microbiome Project data. One of our test cases was in vaginal microbiota, where we knew from previous analyses that there were usually 5 distinct clusters. So we performed the same analysis, and we saw with prediction strength that actually we did get a peak at 5 clusters. It was about the same, so there were also good scores at 2 clusters, so it seems as though the vaginal microbiomes might be split into 2 broad clusters, which then get broken up into 5 subclusters. A little bit later, I did some more follow-up analysis on this to figure out when it was actually useful to do clustering as opposed to doing direct analyses related to gradients in your data or known groupings of your data. And so what I found is that, first of all, if you simulate data with only dominant taxa, with 3 dominant taxa, so this would be kind of an approximation to the Bacteroides, Prevotella, Ruminococcus, 3-way gradients that were seen in the original clustering paper, then the supervised ordination looks really good. So this is where we do clustering. The clustering is done with this algorithm called partitioning around medoids. It's in R. It's the same method that has been used in a lot of the other clustering analyses. And so you do the clustering, you do the supervised ordination where you tell it, I want you to make the clusters look as good as possible using the species values. And this is what you get. Looks like great cluster structure. However, if you ordinate the exact same data in an unsupervised way, so this is just using regular old principal coordinates analysis of Bray-Curtis distances, not trying to artificially make the clusters look better, the clusters are a lot more overlapping, although they still look a little bit separate. But then when you take the colors away, the effect is completely gone. So this is a case where we knew that there were no actual gradients in the data. Following a supervised ordination makes clusters look really good, but the unsupervised ordination, especially when you take those colors away, makes it clear that there is no discrete cluster structure in the data. So this is just a cautionary note about the need to use unsupervised ordination in combination if you're ever going to use supervised ordination, and also to take into account how the coloring might be influencing your visual interpretation. Then we, with the help of Tanya Ward, who's a postdoc in my lab, we did some more analysis of when it's actually beneficial to do clustering versus directly learning a relationship between the species and some experimental outcome. And the reason that we were curious about this is because we know that the microbiome varies continuously from person to person and even within an individual. So if you develop discrete clusters like I'm showing here, the microbiome is continuing to vary within those clusters. And here I'm showing a made-up example where you have 3 dominant taxa, the red bug, the blue bug, and the green bug. And what this means is that, like, let's say, for example, the red bug is most prevalent on the lower right, the lower left part of the plot. Then what that means is that as you get to the beginning of, or the inner part of the red cluster, you have lower abundances of the red bug. And as you move farther down to the lower left, the red bug gets more and more abundant. And similarly, as you move away into the other clusters, there actually is, in this example, there is still some of the red bug. It's just decreasing in abundance as you move farther and farther away from the mode of the red bug abundance. So what this means is that the cluster boundaries are actually being drawn artificially at some threshold, and you're throwing away the additional information of that continuous gradient within the clusters. So let's assume, for example, that the blue bug is a disease-associated bug. And the more blue bug you have, the higher your risk of the disease. So if you were to plot your blue disease risk against the fraction of blue taxon or blue species that you had in your gut, then you should have basically a continuous scatter plot here. And what happens if you first bin your data into these 3 separate clusters, then look for association with blue disease, you'll see that, lo and behold, blue disease is much higher in the blue cluster. Looks great. But you're actually throwing out useful information that could be used to discern between high and low risk individuals. So in this scatter plot, people who were at the far extreme end of the blue cluster, who have the most blue bug and therefore the highest risk of blue disease, they have they're given the same risk score in a cluster-based analysis as the people at the less extreme end of the blue cluster. They're all basically lumped into the same group. Similarly, people who are in the red and green clusters, but kind of close to the blue cluster, they have more of the blue bug than the other people. So you could have people who are on one end of the green or red cluster who are assigned a low risk of blue disease, and people who are on a nearby end of the blue cluster who are assigned high risk of blue disease, but in fact the risk profiles are almost identical because the clusters were drawn arbitrarily. So unless there really is strong discrete structure in the data, we know this can happen with Prevotella, for example. This is a bug that people either tend to have or not have. But for most bugs, they vary continuously from person to person. So unless you have those rare cases of discrete structure, if you're really interested in associating your microbiome with a disease or with some kind of experimental outcome, it's probably better to associate the bugs directly with the outcome rather than throwing out that gradient-based information. Now, to make matters worse, we figured out a way to project a time series of microbiome samples from a single individual onto the some original microbiome clustering of samples. And what we saw was that one person can walk between clusters even in the course of a week. So this person starts in the putative green cluster here, walks down over into the blue cluster and back in the course of a few days. Then their microbiome later on during this 6 months walked from the green cluster over into the red cluster and back. So this is evidence that these clusters really shouldn't be considered discrete. People can vary across them, and that it's dangerous in clinical applications to do this kind of arbitrary cutting and binning of people first, then associating with disease, rather than associating the disease directly with the underlying features in the data. So in summary, I recommend using extreme caution with supervised ordination techniques. So you need to assess the significance carefully. I recommend trying prediction strength, at which point a value of 0.9 or above might be considered significant. This is sort of a philosophical argument. If clustering a subset of your data reproduces the clustering memberships with 90% accuracy, I mean, it's sort of whatever you're comfortable with. There's no rule here about what you can get away with. And if you're using silhouette index, it should be above 0.5, although we've seen that that can be underpowered. I want to emphasize that clusters can be a useful way to analyze high-dimensional data. So if you have 1,000 species, you may not be powered to test all of those species with some disease, but you may be able to find some association of the disease with the overall community structure. And one way to do that is to impose arbitrary clusters on the data and then find out if those bins that you've created have associations with the disease. However, direct analysis is almost always going to be better when you're trying to create some kind of diagnostic test. So a predictive model or a microbiome-based diagnostic test is going to be a much more sensible approach to have the diagnostic test be a function of the abundances of species or genes directly, rather than doing some kind of arbitrary clustering first, and then trying to link the clusters to the disease risk afterwards, after you've thrown out the gradient part of the data. That's all for this video. See you next time.